engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
questions,
Research.
technology.
Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to this week's Naked Scientists. This is the show where we bring you the latest breakthroughs in science, technology and medicine with me, Chris Smith. And coming up, a huge outbreak of dengue fever hits Brazil, so should we be worried here in Europe? Scientists discover the types of microbe that drive the decomposition of animal flesh, including, gruesomely, our own. And where do we get our sense of humour from?
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
Brazil is in the midst of a huge outbreak of dengue fever, with public health experts anticipating a surge in cases across Latin America too. To find out more, we put in a call to James Whitehorn, who's a consultant in microbiology and infectious diseases at Guy's and St Thomas's NHS Foundation Trust. He spent a large part of his professional career studying this particular virus.
Dengue is a viral infection that is increasing in incidence around the world for a number of reasons. But what's been particularly striking is in Brazil that the case numbers are 3 or 4 times higher than they were this time last year. And that's happening for a number of reasons. One of the reasons that's happening is due to the El Nino effect, which is causing increased rain and more water, which promotes more breeding of mosquitoes, which are the vectors that transmit dengue. And because of warmer weather and more wet weather, there's more breeding of mosquitoes and therefore more vectors that can transmit dengue. There are 4 types of dengue virus. There's 1, 2, 3, and 4. And they sort of circulate within the populations over a period of time. But there's been a recent re-emergence of dengue 3 in Brazil as well. And lots of people haven't been exposed to that particular strain of dengue before. And they're being exposed to that, as well as the increased numbers of mosquitoes, which is leading to increased transmission.
Because when people catch a different type of dengue to one they've had before, there's a chance of getting more severely ill, paradoxically, than if you'd never had dengue, isn't there?
Yeah, that's absolutely right. So if you get one infection with one of those strains, you have lasting immunity to that. You have brief cross-protective immunity to the other strains, but once that cross-protective immunity wanes, you do become at increased risk of more severe disease. So having a secondary infection with a different serotype is a recognised risk factor for a more severe disease course.
Do we know why that happens?
The best explanation for this phenomenon is called antibody-dependent enhancement. And what happens in that is that the antibodies that were produced when you were originally infected with dengue bind to the new infecting strain, but they don't neutralise that. These antibody viral complexes are then taken up into cells, and that leads to a higher viral load in the patient who's been reinfected with dengue with a different strain.
Luckily, though, there are vaccines. Are they effective?
There are 2 vaccines that are currently licensed for dengue. There was one which was developed by Sanofi Pasteur, which is the original one which was licensed. And there's more recently been the Takeda vaccine. They are effective, but there are a number of caveats that need to be taken on board with that. So the Sanofi Pasteur vaccine, when it was originally used, seemed to be associated with an increased risk of severe disease and hospitalisation. And when that was looked at in more detail, it seems that patients who were given that vaccine who hadn't had dengue before were more at risk of getting severe disease. Those safety issues aren't the case with the Takeda vaccine, so you can give it to patients who've not had dengue before. And it seems, although there needs to be ongoing work looking at long-term follow-up data, it seems to be safe and effective. And in Brazil, they're using the Takeda vaccine to try and help control this current outbreak of dengue.
And if someone doesn't get vaccinated in time and does get it, what happens to them?
In most cases of dengue, a self-limiting but quite unpleasant infection. So you get a high fever, rashes, you get headache, lots of body pains, lots of aches, and you can be very, very unwell for quite a few days. Some patients do go on to get a more complicated disease course. And the things that you see in that situation are things like dengue shock, when you get leakage from the blood vessels and you get low blood pressure, high pulse, and you need fluid resuscitation. And some patients can go on to get hemorrhagic complications, so they can get bleeding manifestations from the GI tract or from other parts of the body as well.
So you really want to avoid catching this, don't you? If you can't have a vaccine, is controlling the thing that makes the disease spread, mosquitoes, is that an option?
Yes, they are doing that. That's the mainstay of dengue control, really, prior to these vaccines being released. And in Brazil, actually, they're using the army to try and remove the breeding habitats of the mosquitoes from people's houses and things like that. Basically, one of the issues are that mosquitoes breed in standing water that people may have in their homes, or around their homes. So the main vector of dengue is the Aedes aegypti mosquito, which is very, very domesticated. So if you can remove water from plants, etc., in the house, that can reduce the risk of transmission and reduce the burden of mosquitoes. And also you can spray houses and things like that. I mean, that's done widely throughout the world where dengue is transmitted. There's been some really exciting and encouraging work looking at biological control of the disease vector. So there's a bacteria called Wolbachia, which can be introduced into Aedes mosquitoes, and they don't naturally have that bacteria. But when that bacteria is introduced, it can then become sustained in the mosquito population and seems to block their ability to transmit dengue. So the mosquitoes that are infected with Wolbachia will bite people who've got dengue, but they just won't be able to generate an infection and become infectious themselves. And that's been some very, very encouraging work done from a big cluster randomized trial in Indonesia that was published a couple of years ago, which showed that it reduced the dengue incidence by 70% in areas of the city where this approach to vector control was used.
I suspect that many people listening to this who don't live in Brazil might well be in Europe or travelling to Europe. Are we at risk here? Could we see these sorts of diseases as the mosquitoes move, them taking the disease with them and placing us at risk in countries where we were previously pretty complacent and thought we were beyond harm?
Yeah, that's a really good question. I think there has been transmission of dengue in European countries, but they have been relatively small clusters of cases. So in France and Italy, and small transmission events in those settings. There's been a number of kind of climate change predictive models, and looking at the more extreme versions of those predictions, it is likely that dengue transmission will occur more widely in Europe over the next 100 years. But at the moment, I think we're not seeing anything beyond these relatively isolated clusters of cases.
Jamie Whitehorn there. Researchers in the United States have identified what appears to be a network of around 20 different types of microbe that universally drive the decomposition of animal flesh, including our own. It's thought the discovery could have huge implications for forensic medicine, and it was by studying the decomposition of human bodies in specialised facilities built for just this purpose in America that led Jessica Metcalf from Colorado State University to identify these key microbes.
So in the United States, we have these forensic anthropological facilities. And these are places that definitely have really big fences around them. They're outdoors, and where people can choose to donate their bodies to be part of a forensic science or sort of anthropology bone collection, right? And so we coordinated across 3 of these facilities to decompose 36 human donors over 4 seasons. And we sampled these donors who were placed outdoors to decompose daily for 21 days. So these were swabs that we either rubbed a little bit on the skin, or we also sampled the soil right near them. And so we were sort of capturing on those samples the microbial communities that were active. And then we were able to get the DNA out of those, and we used the DNA then to profile what microbes were there at that time. We build that up, and we end up with a time series of snapshots over 21 days for each of those 36 cadavers. And that's a really powerful data set, because that allowed us to see what microbes were there and what they were doing. And also we looked at some of the small molecules they were making as they broke down things like fats and proteins that were coming from the cadavers.
Are you testing different environments and also different times of year? Because I would think that might make a difference to these outcomes, wouldn't it?
We sampled bodies that were placed at 3 different locations that covered 2 climates, and then across all 4 seasons. And what we found is that actually there's this core set of microbial decomposers that show up regardless of all of those variables. And when we started looking into, well, what is the reservoir? Where are they hanging out? Where are they persisting when there's not something decomposing? And we found no evidence for them being, either gut or soil, which were kind of our 2 big environments that we thought, like maybe people were carrying them around. We also looked at these big data sets that are available on the human gut, human body, and many soils. And we really find very little evidence. There's like 1 or 2 of these microbes that appear that you can find sort of in the soils or associated with humans. But for the most part, the only place we found them were on insects. And so what we think, and this makes a lot of sense, if there's animal, it could be a deer, it could be a bunny, whatever decomposing in the forest, that particular location may not have had an animal decomposing on it for 1,000 years. So how do these decomposers microbes persist? Well, they're hanging on to the blowflies and the carrion beetles that can move to visit bodies. And so to me, ecologically, it really made a lot of sense. And previously, we'd known that a couple of microbes that are involved in decomposition came in on blowflies. And now we know it's a lot more than a couple. It's the main key decomposers. Pretty much all of them are associated with insects.
That's fascinating, because we always used to assume that your own bugs just declared war on you as soon as your immune system stopped when you died, and that you were consumed by the army from within. So what you're saying is actually that isn't the case, and it's all an invading army that comes on the feet of flies.
We have a study going on right now of indoor bodies, and so we'll see, because it doesn't mean that these core decomposers are doing all the decomposition, but they're certainly a network that's doing the bulk of it, and they're specialists at it, so they're really good. So some of your microbes could still be betraying you after death and going after your nutrients, but for the most part, no.
How does this change our understanding forensically in that case, given that you've now found this and you've found these species come in, you know the timeline? How does this enable us to have a better understanding of what goes on if someone is killed or dumped or whatever out in the wilderness?
So the cool thing is that because there's sort of these microbes that are everywhere, we can build this fairly general model from which we can predict how long a person's been dead based on the microbes that you sample when the body's found. And we can do that fairly accurately. So this provides another tool in a death scene investigator's tool set, which, depending on how long a body has been decomposing, there may not be much tools available to you. So in some cases, this could be the only one.
Jessica Metcalf from Colorado State University.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound, perfect music for audio and video productions.
You're listening to The Naked Scientists with me, Chris Smith. And I've stepped out just temporarily. I'm actually in the deer park, which is at Peterhouse College in Cambridge. No deer here these days, haven't been for about 100 years or so, but animals similar to deer, their relatives, reindeer, were thought to be being caught thousands of years ago by our more primitive ancestors using stone walls. And an incredible discovery has been described in the journal PNAS this week, where researchers have discovered, submerged 20 metres down in the Baltic Sea off the coast of Germany and south of Denmark, a long wall structure that they think might have been used for this purpose. And with me is an expert on our more primitive ancestors, Emma Pomeroy from the University of Cambridge. She's an archaeologist. What have they actually discovered, do they think, Emma?
So what we've got is about 1 kilometre long, sort of very small wall, if you like. It's not a wall where the stones are kind of tightly put together, but it's punctuated with some bigger stones. So you've got sort of bigger stones and then a collection of smaller stones joining those up. There's more than 1,600 stones, and the interpretation that the authors put forward is that this was a wall that was used to help limit the movements of migrating reindeer so that they could be more easily hunted and kind of picked off.
It's 1 kilometre or so long. It's quite big. So someone made it with a purpose. They speculate maybe hunting animals. Maybe we can come to that in a second. How was it found in the first place if it's 20 metres underwater?
So there is a whole sort of sub-discipline of underwater archaeology, and this involves obviously surveying what's underneath the water off the coast or in lakes. And what they've done is used technology that can kind of tell you what's down there, sonar, and in doing so, in trying to map this region of the sea floor, they've noticed on that there's this kind of linear structure, so a straight line, that doesn't look at all natural. And so then they've gone and investigated further as to what this could be and who might have built it.
When does it date from, and why is it 20 metres underwater?
So it probably dates from around 9,000 years ago, and it's currently underwater because sea levels have changed over the years, and so parts of what was once dry land is now inundated with water. This period, sort of about 9,000 years ago, it's during the Holocene. The Holocene is characterised by much more stable environmental conditions. So the temperature, the plants around you are all much more predictable. And associated with this, we see some interesting changes in people's behaviour. So this is what we would refer to as the Mesolithic period. So it follows the Paleolithic, the Old Stone Age, and precedes the Neolithic, which is what we associate with farming and agriculture. But it's this early period of there being sort of quite a stable climate and stable environment, and we see some real creativity in how people are gathering their food, gathering their resources. In some places we see the beginnings of sort of more complex societies. Some places people are able to be a bit more settled. But yeah, some really interesting kind of specialisation to local resources and innovating about how they're gathered.
How would they have used this structure then? What was its purpose?
So that's a bit of a tricky question, but it's one that I think the authors of this paper make a good case for. So they argue that this long structure went along the edge of a bog or an old lake, and what it effectively did, as herds of reindeer were coming past, they would be channeled, if you like, between this low structure and the bog or the water next to it. And basically it controlled where they were going, and so it makes it easier to hunt those animals, to pick them off, which you can then make use of the meat, the hides, and all the products that come from them.
Do you think it's significant that around the time that this is thought to date from, agriculture is just getting started? So is it a prelude to the way we started to do farming and husbandry, or is it that came along and displaced the need for things like this? People just built rectangular fields and stone walls and kept their animals in there and they didn't have to hunt them.
So I think the evidence we have is that agriculture and animal husbandry is actually introduced from elsewhere. So in Europe, it's mainly coming with peoples moving from Southwest Asia, or what we might call the Middle East. But I think it is significant that it's perhaps linked to this same time period, because the environmental conditions and climatic conditions of the Holocene, where you've got much more stable, much more predictable climates and environments, probably play a role both in the development of these kind of new technologies, but also in the development of agriculture, perhaps a little more recently than this.
Emma Pomeroy talking to me at Peterhouse College in Cambridge. We've all had fun teasing our family and friends in the past. Now, such jokes will often be verbal, but scientists have also observed this behaviour in children as young as 8 months old, and that suggests that language isn't an essential element of this type of teasing. For example, it can be a playful tap on the shoulder. And in light of this, cognitive biologists have been seeking an origin for our sense of humour among our closest animal ancestors. And they've observed teasing behaviours in all 4 great ape species, and that means it's very likely to have been in our biology for at least the last 13 million years or so, as Erica Cartmill, who's at Indiana University, explains.
I was studying gestural communication in orangutans. And I was watching this interaction between an infant orangutan and her mother. The infant orangutan was dangling over her mother. Mother was lying on her back in the straw, and the infant had a stick in her hand. And she reached down the stick and sort of waved it over her mother. And as her mother reached up her hand, started to grab it, the infant pulled back the stick. That kind of, here you go, haha, just kidding, right? Give me 5, too slow. And the infant kept doing it. She did it several times. The mother was playing along, didn't seem to get annoyed, kept sort of humoring the infant. I thought it was just absolutely charming. But what really fascinated me was when the infant stopped doing it, she dropped the stick. The mother then picked up the stick and started doing the same thing back to the infant. I really thought about it as having almost the structure of a joke. There's the setup, I'm offering you something, and there's the punchline. I pull it back, right? There's the expected behavior, take this thing, and then the unexpected, just kidding. And so that formed the center of the kind of thing that I wanted to study in apes. I wanted to understand, was this something that they did a lot? Was this something that where they understood what it meant for another to be surprised.
And obviously, it fits in that group, which we haven't shared an ancestor with some of the species you're mentioning for millions of years. This would argue that some of the behaviours we see in us, the reason we play with our children the way we do, is exactly the same.
Absolutely. I wouldn't argue necessarily that the behaviors themselves are biologically inherited. I'm not saying that we play peekaboo in the way that we do because of our biology, because of our genes. But I think we do things like play peekaboo because we do share underlying ideas about other individuals, what it means to interact with them. We understand that other individuals can have beliefs, they can have expectations, they can be surprised. You really need to understand something about their mind. And I think that those are shared across all of the great apes, those abilities.
How did you test that out, though, to confirm for yourself that that was the case and that this is what's going on? This is a sort of primitive form of humour that has educational value.
Honestly, this is such a scientist answer, but it's something where more data needs to be collected. I mean, it's something that I think we're still exploring. I'm kind of going out on a bit of a limb here and saying I think it's because of these kinds of understandings about other people, other individuals, that we see those behaviors. It's very hard to conclude that when you're just looking at behavior. You can't interview an orangutan and say, what do you understand about this other individual? You need to draw on information from a wide range of sources. You need to look to see what they do spontaneously. You need to try to design clever experiments and games where we can see what do they pay attention to, what information do they use, how do they make judgments in one situation versus another in order to really fully answer that question. What we were doing in this study was kind of laying the groundwork for that. What we started with was one example, or a handful of examples of what I had seen orangutans do. And I wanted to build a corpus of behaviors. I wanted to build a data set. Do they all look like this? I offer something and I pull it back at the last second, or are there other kinds of behaviors that might have similar underlying structures but appear quite different on the surface?
Do you think this goes beyond just higher animals like these primates that you're describing, though? Because my dog turns up with a toy and will torment me because he'll wave it in front of me and then whip it away before I have the chance to take it off him. And it's clearly a game. They clearly are getting something out of it. Why would other animals be doing that?
I think that's one of the things that we hope will come out of this study. This isn't the first time anyone has seen these behaviors. You go to a zoo and you watch apes, you'll see some of them. And, it'll probably be familiar to pet owners or people that work with or watch other animals. The thing that really we were trying to do is to argue that there isn't a sharp division between aggression and play, that there's an interesting gray area in between where what you do can be interpreted as playful or it can be interpreted as mean. And I think learning to navigate that space, learning what's appropriate with which individual when, is something that social animals in particular have to figure out in order to thrive in their groups. And this is something that we know humans do. Now we know great apes do it. And so our hope is really that by sort of shining a light on this kind of behavior and defining it and describing it as its own thing, we will encourage other people to look at it in a different way and to really start paying attention to it. And we'll start to collect examples, similarities and differences across a wide range of species.
Erica Cartmill at Indiana University there. Well, it's time now to join James for our question of the week, which is sponsored by Clam XAV, the anti-malware software that protects your Mac without compromising performance. Tried and tested by us, Clam XAV provides top-quality malware and virus protection for your Mac that won't slow down your computer. And even better, listeners to The Naked Scientists can get 20% off the first year using code NAKED20. Go to clamxav.com/naked and use the discount code NAKED20 to protect your Mac today. James.
Thanks, Chris. This week, we're reflecting on this question from listener Lotus. If I wear mirrors all over my body when it's sunny, would that keep me cool? Thank you, Lotus. The short answer is yes. Mirrors are very efficient at reflecting sunlight. When sunlight reaches the surface of the Earth, over half of its energy is heat. So a mirror placed in the way of your body will do a good job at reflecting most of that heat. But glass is not necessarily the most practical material to be making clothing from. It's not very flexible and can cause irritation to our skin. Clothes made from white textiles, while less efficient than mirrors, will reflect more sunlight than darker clothes, which will keep you relatively cooler, and it's why, for example, cricketers wear whites. But Stanford University's Shanhu Fan has produced a special brand of silk to mitigate the effect of ultraviolet radiation from sunlight warming up this comfy clothing material. Here he is speaking to us a few years ago about this work.
Native silk absorbs ultraviolet light. Absorption of ultraviolet light gives rise to heating of the textile. We introduced aluminum oxide nanoparticles into the silk. Nanoparticles just look like a little ball, except they are very, very small. The nanoparticle that we put in is designed so that it reflects strongly the ultraviolet light.
And Professor Fan has proven the effectiveness of his special silk at keeping us cooler.
What we have demonstrated is that nanoprocessed silk, when placed under direct sunlight by itself, can reach a temperature that's 3.5 degrees Celsius below ambient. And therefore, a person wearing such a nanoprocessed silk in an outdoor setting is essentially wearing its own air conditioning.
Not bad, eh? The nanoparticles are locked tightly in the silk with the use of a chemical bonding agent, meaning they don't come out in the wash. So Lotus, yes, mirrors might keep you cool out in the sun, but there are perhaps more practical solutions out there, aside from seeking shade and drinking lots of water, of course, in the form of white clothing and nano-processed silk. Thanks for sending that one in. Next time, we'll be answering this question from listener Ranjith. He says, I've been seeing some claims that volcanoes produce a huge amount of CO2 compared to CO2 emissions by humans. Is this true?
Thank you, James. And if you've got a question for us you'd like us to get our teeth into, why not send it in? It's chris@thenakedscientist.com and we will see what we can do. That's all we have time for today, but do be sure to tune in on Tuesday when we'll be taking a closer look at forever chemicals and their partners in crime, the microplastics. The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education. It is supported by Rolls-Royce. I'm Chris Smith. Thank you for listening. And until next time, goodbye.